So today's webinar is dealing with finding the best structure for what you need in the PDB archive. My name is Matthew Conroy and I am a data curator here at the Protein Data Bank in Europe. If you're unfamiliar with the Protein Data Bank archive, then we're an archive of experimentally determined three-dimensional structures of biological macromolecules. And the experimentally determined is critical in the PDB archive. All our data come from a sample which has been in a tube at some point. There's no theoretical data that we have. And while we're called the Protein Data Bank, and most of the data is indeed three-dimensional structures of proteins, there are also nucleic acids, complexes of proteins and nucleic acids, and there's a few sugars, and of course small molecules that bind to proteins, drugs, inhibitors, cofactors, etc. in there as well. The Protein Data Bank is managed by a consortium of different organizations across the world together called the Worldwide Protein Data Bank. And we coordinate data that arrives in the PDB from across the world in the different sites. So ourselves here in Cambridge in the UK, we deal with all experimentally determined data that's solved in labs across Europe and across Asia. And together with our partners in the rest of the world, we work together to put data in the PDB, which is the FTP archive of data files. And then from that archive, we serve that common data out on our respective websites in different ways with different search systems, with lots of value-added information to help you find what you need and use the best data in the easiest possible way. It's important to realize that whatever the size of the molecule in the PDB, from the very small molecules, for instance this copper scavenger, which is less than a kilodalton inside, to the huge complexes such as Zika virus, all the models in the PDB are interpretations of experimental data. 88% of the data roughly are solved by X-ray crystallography, and the data that the model is an interpretation of is the structure factors, and it's been mandatory to deposit the structure factors since 2008. 10% is solution NMR, and the data there are the restraints, so saying which atoms are close to which other atoms, and the chemical shifts, which we'll talk about later, which is saying the frequency with which the atom resonates in the magnetic field. And it's been mandatory to supply those to us since 2008 and 2010 respectively. And 1% is electron microscopy, but this is growing greatly with the new advances in electron microscopy. You may be aware the Nobel Prize this year was awarded for EM, electron microscopy techniques. And the data deposited in the electron microscopy data bank has been mandatory since last year. So while the PDB as an archive of protein models has existed since 1971, the backup data from which those models are derived has been deposited in the archive relatively recently in that timeline. And the mathematicians among you may notice that those percentages don't add up to 100. There is a small percentage by other methods which we won't be talking about today for reasons of time. But given that data, and as in all science, some data are better than other data, and some models, so the atomic models which are interpretations of that data, are better than other models. So here's some data. There's two photographs of myself. One of them is clearly recognizable, and the other is rather poorer data, and if the image on the right was CCTV in a bank and I'd robbed that bank, then I'd probably fancy my chances of getting away with that bank robbery. Here's two interpretations of that data, one of them rather more flattering than the other, but it's important as well to realize there that the image on the right, if all you need to interpret from the data is that I am a white male human, then that data is perfectly sufficient. And the same as the data in the PDB, you need to realize what interpretation is being made from the data. For whatever you need to do, you need to find the best model. You need to be aware of potential limitations in that model, because different techniques have different strengths, different models have different features in them. There's a very good paper in Trends in Biochemical Sciences, which came out earlier this year, which gives you an overview of the strengths and limitations of the different methods, and points out structures are not absolute truths. They're models that fit the data. There's uncertainty and there's subjectivity with all the models, and this tells you where the pitfalls are, what to look for, and what the limitations are. So now we'll ask you, before we carry on, what do you use the data in the PDB for primarily, so that we can assess what you might need to get out of the database. Okay, so around half of you are using the PDB to find a template to make homology models, and a further third of you are looking at protein-protein complex prediction or small molecule docking, and the rest are using it molecular replacement, so you're presumably crystallographers, or to explain biochemical information, or for something else, the enigmatic something else. So, as we've said, it's important to know what the limitations are. All structures in the PDB are models explaining the data, and we don't reject any structures based on quality. So, if a model is truly terrible, we can't say this is too bad to go in the archive. We can, however, give you an indication of how good or poor the model might be. And this guy, George Box, said that essentially all models are wrong, but some are useful. He was talking about statistical models, but a similar thing holds true for atomic models. All models are incomplete interpretations of the data. So what are we doing as the worldwide PDB to indicate how good the models are and the data? Well, we have validation task forces from the structural biology community that have been commissioned, and they've advised us on what we should do to determine the quality of data and the quality of the models that fit to them. And these task forces published papers that are all in the journal Structure, and they're there on the left, which detail what they concluded, and they advise us how best to validate the models in the archive, the experimental data, and how well the model fits with the experimental data. So that means now that most entries in the archive come with a validation report which gives you that information. It comes in PDF, which is human readable, and there are XML formats for machine reading, and it's also available programmatically via our API. So for structures determined by X-ray crystallography, you can see that we assess the model quality, the data quality, and the fit of the model to the data. For NMR, we do the same things, and for electron microscopy, we assess the model quality. We don't yet, as the worldwide PDB, assess data quality or fit of the model to the data, but as we'll see briefly later on, that data is available on the AMDB pages here at EBI. This is what the PDF of the validation report looks like. This is the front page, and it's available on all the worldwide PDB sites, and we recalculate it annually because, as you will see in a little while, we assess the quality relative to other structures in the archive, and because more and more data is added, because your position relative to the other data changes over time. It's also available at deposition, so if you solve a structure and you deposit it with the PDB, we supply a report at the time of that, and that's for the authors to look at and hopefully to go out to referees when the paper is being published, so the referees can assess the quality of the data. And there's a standalone server too, at that URL validate.www.pdb.org, and you can put any model and any experimental data in there to assess what we will say about it when you submit it. So whether that's a homology model, you can just look at the quality of the model, or you're refining a structure and you want to know how you're doing, then you can upload your files to that server and we'll give you a validation report. On PDBe's pages, they're available from the search results. So this is a search for HIV protease or HIV retropepsin, and if you go to this link, download files, and it opens up a pop-up box. There's lots of files you can download. One of the sections is validation, and there we'll see the summary PDF reports, the full report, which is a longer version, and the XML, and a couple of graphics as well, which I'll talk about shortly. Similarly, on a particular PDB entry page, they're available for download and viewing. So the view and download tabs there. There's the report, and there's the XML to download, and the download. So view will pop it up on the screen, and download will download it for you to look offline. What does the report look like? It starts with a summary, and as you go through the report, it gets progressively more and more detailed. So it starts with overall quality at a glance, and here you see across a variety of metrics where the structure ranks relative to other structures in the PDB archive. Towards the blue is the higher end, the better end, and the red is the poorer end. There are then residue property plots for all the macromolecules in a PDB entry, and they highlight outliers in different colors, which we'll talk about later. And there then follows a detailed analysis of any potential issues, which may run to several pages, and if it's a very large structure, many pages. So the overall quality at a glance looks across a variety of different measures how well the structure compares to others in the PDB archive. It's important, though, to realize that poor ranking doesn't have to mean that the structure is wrong. The nature of maths is not everything can be above average. It may just mean it's not as right as others, and there are justified reasons for residues being outliers. So there are very good reasons why things can be odd. The important question to ask is, are those outliers, are those oddities supported by data? And we use this information, this summary, to rank results at PDBe. So on our search pages, you'll see these two slider bars on the right-hand side above each entry that indicate the quality. And this is a summary of that information, which allows you to assess structures which have the fewest outliers, and you can sort the results by those quality metrics. So what do the summaries tell us? So the quality of the geometry, the quality of the chemical structures in the PDB is assessed using software called Molfrobity, and that's used to compare a structure to others in the archive. And irrespective of the method used to determine the structure, whether it's X-ray, NMR, or electron microscopy, they're all judged the same way. This is good because chemistry is chemistry irrespective of how you look at it. So one of the metrics is clash score. So this is atoms bumping into each other. We know that two atoms can't be in the same place at the same time, so if two atoms are overlapping, then that highlights that there might be a problem. The other three metrics, Ramachandran outliers, side chain outliers, and the RNA backbone, indicate surprising bond angles. We know from the many structures that we have in the archive that there are some typical conformations that we see for amino acid and RNA residue geometry. So anything comes outside that value will flag that as a surprising result. And you can see there's a filled in bar and an open bar on there. That's compared to, in this case, the full bar is all structures in the archive, and because this is a structure determined by nuclear magnetic resonance, all other structures determined by the same technique. For X-ray crystallography, it's similar structures of the same resolution, or similar resolution. Of course, good geometry doesn't have to mean it's right. And I see lots of people doing homology modeling that assess the geometry of their model, and the geometry is fine, so they conclude that their homology model is correct. Good geometry tells you that your model is chemically sensible. So this model here on the left might be perfectly chemically sensible. But if the protein actually looks like that, then it's completely wrong. So this is a trap that's commonly fallen into and you should avoid. Good geometry just means chemical sensibility, and it doesn't mean right. For X-ray structures, there are another couple of metrics on the summary sliders. You'll see at the top and nearly at the bottom R3 and RSRZ outliers. R3 is a crystallographic measure which tells you how well the model back predicts the data. So if you take your model and say, given this structure, if I calculate the data back from this as theoretical back prediction, then how well does it agree with the actual data? And lower values are better. A value of around 0.2 in this case is reasonable and what we'd expect. Values between 0.15 and 0.25 are typically okay. And the RSRZ outliers are residues which are not in electron density. So these are atoms or amino acids in the structure which do not fit into the data from which the model has been built. So RSRZ is a Z-score of RSR. It measures the fit between the amino acid or the nucleic acid and the data. And the Z-score is a normalization of that, particular to the residue type and the resolution bin. So we ask the question, how well does your arginine fit the data? And then, well, how does that compare to all other arginines at a similar resolution? And a residue is flagged as an outlier if the RSRZ is greater than a value of 2. So we've talked about X-ray data a lot. Let's have a look at some X-ray data. From the diffraction pattern, which is what you get directly by shining a beam of X-rays at a crystal, the map can be calculated. That indicates the location of the electrons and therefore the atoms in the crystal. Hence, it's called an electron density map. So this is what an electron density map looks like, and into that map you build the atomic model. Model building is an iterative process. You need to know roughly where the atoms are in order to get a sensible map, and as the model gets closer to the data, the map improves. So you have to build it iteratively, but then you can assess how well the model fits the map. And the resolution of an X-ray structure indicates the precision with which you can place the atoms. So here we have several images of a heme group at different resolutions. 3.7 angstroms here. Well, you can sort of see that there's a blob that the heme fits into. It may be the other way up. It's hard to tell, but you can sort of see where it should be, but no more detail than that. Better resolution at 2.5 angstroms. You can see the overall shape. I'm now convinced it's the right way up. 1.5 angstroms resolution. This is really quite good, and you can model with a lot of confidence where the atoms are. You start seeing holes in the rings. And get to better than 1 angstrom resolution, and you can put in with a lot of precision precisely where every atom is. It's important that in low resolution data, an atomic model might have been dropped in order to indicate just the location or the orientation of proteins. So this is an electron microscopy map at 37 angstroms resolution of an FDPA and ATPAs, F1, F0 ATPAs. And the purpose of building the model into this map was simply to show the location of the proteins and that they fit the data. The conclusion of the paper was basically this angle. So the model was perfectly justified to say the data is consistent with these being ATPases and they sit at this angle. If, however, you use this data in order to measure a hydrogen bond within a helix, then you may be extrapolating rather too much from the data that's there. And that's an important question to ask when you're looking at data, is what is the original conclusions of the model that the authors gave, and are you concluding rather more than was intended? So let's look in more detail then at the validation report. We next move on to the residue property plots, and they're given per polymer chain. So they're color-coded by residue according to the number of geometric outliers. So if a residue is green, there are no outliers. Yellow, there's 1, orange 2, and if it's colored red, there are 3 or more outliers. And if it's gray, then it's not there in the model. We know some residues are not built into particularly X-ray data because they're mobile and they can't be seen. In this case, there are no unmodeled residues in this example. And those outliers are listed in the metrics that I've given on the right. Bond lengths and angles, chirality issues, too close contact, so clashes, Ramachandran and side chain torsion angles, so peculiar torsion angles, and RNA sugar pucker. We know typically what the sugar should look like in an RNA, and if they're out, then that's surprising. But this color coding is important to remember because we'll see that later in various other different views of the structure that we see, which keep to the same color coding. At the top is a summary bar chart. So there we see that 63% of the residues are green, so they have no outliers. 34% have just the 1. And then there's a small percentage, which isn't listed there, that only have 2 outliers, and none have 3 or more. The red bar over the top is equivalent to the red dot that appears over the top of those summary charts, and that's telling you the percentage of residues for which there's a poor fit to the electron density. That's the RSRZ outlier that we talked about earlier. So the red dot there indicates which residues don't fit particularly well to the experimental data, and in this case it's 7% of them. On the PDBe website you can see the validation. There's the summary there, there's the sliders that occur, and in our 3D visualization you can also see the validation information. So there's HIV protease, you can rotate it, go to the validation report options, hit apply, and now the molecule is colored according to those geometric outliers colors we mentioned before. Green, no outliers, due to red, 3 or more. Click on a residue and then you see the electron density, so you can judge by eye whether or not the atoms fit the data adequately or not. Similarly, if you scroll down to the molecule details page and click on molecule details, you get a more detailed view. So there we see information in sequence, in topology, and 3D structure. So there's the validation report going along the sequence, and if you hover over a residue, it tells you what the issue is with it. And the red dot there tells you it's an outlier. The topology diagram you can also color by the validation report. This is in the same way, and hover over a residue, it tells you the issue with that amino acid. And as we've seen just previously, you can color the 3D diagram by the validation report as well, and all these 3 views are interactive. So if we hover here over that methionine, this tells you it's an RSRZ outlier. So the red dot indicates it might be a poor fit to the electron density. Click on it, and it zooms in the 3D, and click on it in the 3D, and that shows the electron density. And there we see there is no electron density for that methionine. So chances are that that amino acid is mobile in the crystal structure, so the electron density is blurred out because it's moving. So while it's certainly there, you can't be confident of the position of the atoms at any given time. You'll also have seen that on the 1D there's a different tab, the Uniprot coverage viewer, and this gives you a view for a particular protein of the different structures in the PDB and which parts of the molecule they contain. So if for you the best structure is not necessarily the best quality one, but the one that contains most of the protein, then this is an interesting view to look at, where you can see whether there are fragments of the protein or the full length protein. So we've looked at X-ray data. Now let's have a look at what we do in the validation reports for NMR data to assess the data quality and the fit of the model to the data. It's much less comprehensive than X-ray data is. We firstly give the completeness of the resonance assignments, the percentage of the atoms in the model for which a chemical shift is measured. The chemical shift is the frequency that the atom resonates at in a magnetic field. Every atom resonates at a particular frequency when it's put in a magnetic field. How many of those have we worked out what the frequency is? And then for those, we give those shifts which are statistically unusual. Again, there may be a good reason for it to be statistically unusual, but it's worth looking at. And then for the fit of the model to the data, we look at the random coil index. So this asks the question, does the chemical shift and the conformation of the protein agree? We know, given the chemical shift on an atom is influenced by its secondary structure, by its position within the protein, and whether it's ordered or not. So we ask the question, do these two things match up? And there's a graph here in the validation report. Residues which are disordered in the model ensemble, so they're mobile, are coloured cyan, and residues which are well ordered are coloured black. And the x-axis here is the sequence of the protein. The y-axis is the random coil index, which tells you the likelihood of disorder given the chemical shift of the particular residue. So the take-home message is that cyan bars should be tall and black bars should be short. If it's the other way round, that might indicate that there's a problem with the model and the fit to the data for an NMR entry. For electron microscopy, the data quality and the fit to data is not yet in the validation report. It is, however, available in the electron microscopy data bank here on EBI's pages. And for reasons of time, we won't go into this here, but perhaps the guys from EMDB can give a webinar in the future looking at EM data and fit to EM and fit to models if you should wish it, and I'm sure you can give that in the feedback if that's what you'd be interested in. So the geometry of the model and the fit to the data go hand in hand. Look at this example, valine 48 in residue 3KSE is a Ramachandran outlier, and it's flagged as an unusual residue in the validation report. But the strained conformation is well supported by the data. It fits the electron density well, and biochemically we know that this residue is strained. It's been well characterized. In the same entry, aspartate 62 has some strange bond lengths, and looking there at the electron density, there is no data to support those unusual bond lengths. So in the absence of good evidence to conclude why it's unusual, I think it would be fair to assume that those bond lengths are probably incorrect. And the model, because there's poor data, has actually just wandered away from normality somewhat, because they're not justified by the data. A brief aside on assemblies, because a lot of people get this wrong. For crystal structures, it's important to realize that only the smallest part is deposited in the PDB archive, and the whole can be generated by applying symmetry to it. So the part you're interested in could be the file as it is in the PDB. It could be the file plus symmetry to make more of it, so apply symmetry to that. Or it could be only part of the file. If this is the whole file, you might be interested in only that bit. HIV protease that we've been looking at throughout this webinar, in this entry 2AZ9, that's the deposited file. It's only a monomer. But HIV protease is active as a dimer, and the substrate and inhibitors fit in the middle there. So if you're docking potential drugs into HIV protease and you're docking them into that, then you're on a wild goose chase, because that's not the form of the protein that's active. You need to look at the whole, and these are annotated in the PDB. If you look at the assemblies section, this clearly indicates what we think the assembly is. That was for macromolecules. How do we validate small molecules in the PDB? Like for macromolecules, there's a summary section in the validation report, which gives you a summary of potential issues. So the chirality section, is the handedness of the molecule correct? Is it chemically sensible? And we know how long a carbon-carbon bond should be. In this case, is that right, or is that way off? Does it hit anything? The clash score, is it banging into anything? And for X-ray crystallography entries, does it fit the data? There are thousands of amino acids and nucleic acids in the PDB. They're well characterized. We know all there is to know about an arginine geometrically. We know all there is to know about an adenine. But your small molecule could be unique. It could be the only instance of it in the PDB. So how do we tell what that should look like? Unfortunately, we can use MOGL from the Cambridge crystallographic database, and we use MOGL to compare bond lengths and angles with chemically similar fragments. So their database contains thousands and thousands of chemicals, and we can look at the molecules and say, this structure is unique to the PDB, but it's similar to other stuff in the CCDC. So we can plot a graph and say, for similar fragments, this is the distribution of bond lengths. So if our molecule is over there, that's somewhat surprising. And again, this is in the validation report. Bond lengths and bond angles and strange values are flagged. The fit to data, however, we have the real space R value, the RSR, exactly the same as we do for macromolecules, and the real space correlation coefficient, which are both measures of how well a residue fits the electron density for X-ray structures. RSRs above 0.2 or RSCCs below 0.8 means it's worth a second look and asking the question, can I be absolutely convinced of this data? A further metric, LLDF, assesses how well the molecule fits relative to its nearby polymer. So how well does it fit the binding site? It's by no means a perfect metric, but it's what we have in the validation report at the moment. So it asks the question, is the ligand data comparable to that of the binding site that the ligand's sitting in? And you can see here for this particular one, the ligand at the top, DIF, which is the painkiller diclofenac. The LLDF is vastly greater than 2. The RSR is much higher than 0.2, and the RSCC is much lower than 0.8. So all those metrics indicate that there may be a problem with this ligand. And when you look at the electron density for it, there is very little. So you may have seen on some of those electron density images that not only is there a blue map for the electron density, but there are some red and green bits as well. Because along with the density map, there's a second map which indicates in red those areas where the model has too many atoms for the data, and in green those areas where the model has too few atoms for the data. So the data in green suggest that it wants more atoms in that part of the model, in that part of the map, and the red bits say there are too many atoms here, I don't want them here. In the case of this tryptophan, I hope you can see there's a very easy solution to this. It just needs turning round, and then it all fits marvellously well. But the difference maps are useful and instructive to look at sometimes. So for instance, this structure is a hint of something maybe bound to the heme in this cytochrome P450. Difficult to tell, their maps are noisy, so it's hard to tell, but you might start asking the question, should there be something there? In this one, there's a huge green blob indicating I want more electrons here. And actually in the paper for this entry, the authors say, we see electron density, we think something should be here, but we don't know what it could be. Red density, on the other hand, suggests that there are atoms there that shouldn't be there, and in this case, this aspirin is not supported by the data. So the data is saying, I am unhappy with these electrons being modelled here in the model. There's also a clue that this aspirin molecule isn't actually interacting with the protein much, so you can't really tell why it's there because it's not bound to anything. Each ligand on the PDBe pages has its own page. So if you click on the ligand here, 3TL, you can see lots of information about the ligand at the top, and then scroll down a bit. You have a 2D diagram looking at the binding site, and you can see the binding site in 3D, complete with the electron density for X-ray structures there. So you can assess the quality of the ligand and how it fits the data, and how it sits in the binding site. So to summarize what we've said today, I hope you have an appreciation now that it's not a black and white issue. Structures can't easily be assigned to be good or bad. It's not black and white, it's not red and blue either. But you need to look at the validation report, see how many outliers there are they justified? So a summary slider like this is okay, lots are towards the blue end. If everything was crashing on the red end, you need to be a little suspicious, but somewhere in the middle may be fine. But are the outliers talked about in the paper if there's a paper describing the structure? Does the model fit the data? And does the structure adequately explain what's known biologically? Taking the structure with lots of biochemistry, do they all agree with each other? Do all experiments agree with each other? Then you can be reasonably confident. If the structure and the biochemistry completely disagree, then you have to start asking questions. Is one of them wrong? Are both of them partially right? The worldwide PDB validation reports are extensive, and they're a good aid to identifying good structure quality. And at PDBe, you can search, your search results can be ranked by the quality. So if you have several hundred HIV proteases, you can rank them by quality and get the one which has fewest outliers at the top. And our pages allow the validation to be viewed in 3D on the structure. 3D structures don't come with error bars, but this is the closest we can get to it, showing geometric outliers and the fit to data for electron microscopy and X-ray entries. It's also worth noting that available from the PyMol wiki, there is a plugin that we have to show the geometry, so you can open your structure in PyMol and color it by these geometrical outliers, green, yellow, orange, and red too. So that brings us to the end of this brief look through data quality in the PDB. I have to acknowledge our PDB partners and the guys who make all this possible. And thank you very much for your attention.